it got really quiet in here. Welcome, everyone, to the Dean's Conversation series. Today we are doing bays, bugs, and bacteria, the ecology of survival. So this year our theme in the Dean's Conversation series has been on the sciences. So our first one was on biomedical or chemical sciences. The next one was on astrophysics, and today we're doing ecology and biology. So thank you to our panelists for joining us today. Thank you all for coming out on such a brisk afternoon or evening. So let me introduce our panelists, and then we'll jump right into it. So next to me is Karen Gadan. She is an associate professor of biology with expertise in marine community ecology, tidal, wetland ecology, and conservation biology. Next up is John Lill, professor of biology, systematics, evolution, and ecology, who has an expertise in plant-insect interactions, insect ecology, and evolution. And then at the other end of the table, we have Jimmy Saw, assistant professor of biology and microbiology with expertise in diversity, ecology, and evolution of microbes in extreme habitats. Looking forward to hearing from you on your research. So why don't we get started right away with just this is sort of the elevator pitch. What do you do? So can you give us a brief introduction to your research and help us understand the importance of studying ecology?
Sure. Thanks for having us here. And if you couldn't tell from the introduction, I'm Bays. And so my work takes place in the Chesapeake Bay and the coastal bays of the mid-Atlantic. I'm very interested in how climate change and especially sea level rise is changing our coastlines and changing the natural and human managed ecosystems that are there. So this is interesting because we're a flat coastal plain where even incremental sea level rise is having really big effects, and change is happening right now before our eyes. So as an example, we've lost 200 kilometers squared of salt marsh, intertidal marsh, drowned to the sea in the last about 40 years, since 1984. And we've also had about an equivalent amount of forest, upland forest, that converted to tidal marsh. And so these are the kinds of changes that I study, and I try to get into the mechanisms behind those change and some of the intermediate steps that we see changing in the plant communities, and then use that to help with management practices and information that can help the people that depend on those ecosystems, including forestry sectors and farmland.
Is that on? Yeah, you can hear me. Thanks again for having me. So my interest in ecology really stems from my really deep fascination and appreciation for natural history. So the different ways in which organisms deal with life and confront the challenges that nature poses. And because plants and the insects that feed on them make up most of the described species on Earth, they've had my unwavering attention for most of my career. And because populations of plant-feeding insects, they are also heavily impacted by their own consumers. So things that prey upon insects that feed on plants are sort of understanding, and my interest in this field comes from understanding the joint challenges of finding food and escaping your natural enemies. So next slide. So this cartoon really sums up my research in one very simple cartoon, which shows a caterpillar eyeing a plant, not only for its nutritional value, it needs to obtain and find food, but also for its protective value, because plants often represent more than just food, they're also a habitat for the insects that feed on them. So next slide. And so when you add that third trophic level, the possibilities become endless. So the niches that these insects occupy now are sort of exponentially increased when you add and consider the ecology of these animals from this multi-trophic perspective. And so my lab group conducts field research. So we go out to the woods basically in the area, and try to characterize all the different complex interactions and structures of these interaction networks that occur between plants, the insects that feed on them, and then the natural enemies that feed on them. And finally, just what really motivates my research is this modern approach to understanding ecological interactions that incorporates both ecological and evolutionary influences. So in this little cartoon shows the interplay between ecology and evolution that really shapes the ecology, the morphology, the behavior, the life histories of these organisms. So from this little diagram, you can see there's a little caterpillar being eaten by a wasp. So those interactions, predator-prey interactions, have fitness consequences, which then drive evolutionary processes like natural selection, which then change the distribution of traits that then feed back to ultimately impact the interaction. So it's this endless loop of ecology and evolutionary biology interacting to shape all the diversity that we see in these systems.
Hello, can you hear me okay? Okay. All right, thanks everyone for coming. I'm interested in microbial ecology and diversity and extreme places. So what I mean by extreme is, the environment that may seem really difficult for humans to thrive. We see microbes thriving there. So it's just, looking from anthropocentric kind of view to say. And so I'm focusing on these environments because I'm interested in early evolutional life on this planet. So I'm focusing on two different habitats. The first one is a volcanic environments that you might see on the island of Hawaii. And the second is these terrestrial hot springs. So my lab is currently investigating how life, what kind of diverse life forms we can detect in these surroundings, and then also how they are co-evolving with the environment, how they're responding to these extreme conditions, how are they adapting them. So that's the most fundamental aspects of my research. And so I'm just showing you some of the picture here to show I do quite a bit of field research as well. So I go to these sites and bring my students to the field, and then I collect samples from those locations. So this is a picture of me inside a cave in this volcano in Hawaii. So I'm happy to answer more questions.
Thank you. So my next question for you all is, so what have you found that's unusual or surprising in your research? And we'll start with you, Jimmy. And Karen, I understand that microphone's not working, so we're going to trade off back and forth.
Right. So the surprising thing about these extreme places is that sometimes you may think that, okay, these are extreme environments, but then you see, okay, there must be limited forms of life that can thrive there, right? So, but on the contrary, what we've been seeing here, that there's actually very diverse forms of life thriving in these extreme places. So, for example, if you look at these, the images over here, these are what we call biofilms. So these are basically microbial goo. So they make these sticky substances where they would attract things around the surroundings. They build communities that we cannot really see with the naked eye here. But then when you on the surface, you think it's like greenish, so there must be algae or just some sort of a photosynthetic organism. But if you actually look at this diversity of life using sequence data, you can see that there's actually a lot more diverse lineages of bacteria that you don't see it just from the surface. And then the next slide is also, this is a hot spring that we've been collecting samples from. And then so here as well, you can see such a hot type of environment that you don't expect a lot of things to thrive, where we're seeing lineages of microbes that are really closely related to eukaryotic life form. And then they are really interesting to study. So we don't really expect such diversity of these simpler life forms on these extreme places, but yes, here we're seeing them. So that's one of the biggest surprises I've been seeing so far. Yeah.
That's a really good question. So I guess one of the surprising things is that even though I do my research literally in forests around DC and suburbs, including Rock Creek Park, our lab has discovered several new species to science just in our own backyard. So I work a lot on interactions between a really diverse and poorly known group of insects called parasitoids that attack and use caterpillars as hosts. And we've reared several just from Rock Creek Park that were not known. And in case you don't know, scientists have been collecting insects in the DC area for as long as the Smithsonian's been around, so the collection's rather extensive. So to find new species is always a little bit surprising. The other thing that I could think of that was unusual is one of my graduate students, Ryan, several years ago, she was working on a really common butterfly that feeds in brassica crops around the area. And normally when these caterpillars get attacked by their natural enemies, these parasitic wasps that lay their eggs inside the bodies and use them as a developing host, when the parasitoids emerge, they kill the hosts. And that's the usual thing that happens. And what she was finding is that after the parasitoids emerged, the host was staying alive for sometimes weeks afterwards as a zombie bodyguard protecting the parasites that killed it. So how that works and how that behavior evolved is really, really interesting and the subject of a future grant proposal. So.
Well, in my research, I've chosen to focus on one of the sea level rise transitions, which is when forest becomes marsh, because it's indicative of this sea level rise pattern. And my background is actually in wetlands, in tidal wetlands and the marshes. And so those are treeless environments largely. So I come to forest ecosystems with very naive eyes. And so I've had a lot of surprises, which I think has really benefited our research in doing this, coming with this kind of naive perspective. So I'll tell you about two of these. One of them is that we hooked up these sap flux sensors to our trees to try to measure the water use of trees, because the salt water is very osmotically stressful for the trees, and they can't take up as much water when they're inundated by seawater. So we were trying to track the water use, but we saw that across the forest, all of the trees, when they got inundated, actually showed a reversal in their water flow. So water normally moves from the roots to the shoots. That's your intro bio. But in this case, the water in the trees across the entire forest was moving from shoots down towards roots. So that was a big surprise and a learning moment, and maybe wouldn't have even been caught by scientists who study the forest better, because they tend to use a different kind of sap flow measurement that wouldn't have even seen this kind of pattern. So that was fascinating, and we learned from that these trees actually rely quite a bit on foliar water uptake, meaning they take up water through their leaf surfaces and some pores in their leaves that they can use metabolically instead of roots. That was fascinating. And another discovery that we made in the lab, but we're continuing to research, is how the trees can change their investment in their leaves in order to conserve water. And so we find that has kind of rippling effects into the rest of the ecosystem. When the trees use less water, there might be more water available for other species, and it might slow that change from forest to marsh and have kind of an ecosystem scale effect. So we've been studying this response of the organisms and how that really might change what we see on the landscape, and I've been finding that really surprising and fascinating.
Fascinating discoveries. So All right, so the next question up, and we'll go back down that way on the table. So how has your field changed over the last decade or so, or over the length of your career? For example, are you still trying to answer the same kinds of questions, and have techniques changed?
Yes, things have definitely changed a lot. When I used to study tidal wetlands and people were thinking about sea level rise, people weirdly only thought in the vertical dimension. How much sediment are marshes trapping that might allow them to keep pace with sea level rise? And I think studying this forest transition really allowed people to see that ecosystems shift in the XY dimension as well. And so that kind of put a different paradigm on the response of ecosystems to sea level rise, and it's been pretty transformative. At the same time, sea level rise has been accelerating, and so we started our research at a field site that we thought maybe we'll get lucky and we'll get a big storm and see all this impact. Just a month later, it was inundated for the first time, and it's been inundated over a dozen times since. So, we've learned that moderate inundation in these kinds of ecosystems is actually quite common, but is increasing in frequency before our eyes. And then lastly, I think in my lab, we're using drones a lot more. So the technology aspect of drones and machine learning techniques that allow us to interpret imagery has been quite transformative. And that's the image that you see on the slide behind you is an example of drone footage of an area of forest mortality. We call that a ghost forest, where a combined degree student in my lab is now using machine learning to detect in these images live and dead tree canopies, and we can scale up our research then to the forest scale very quickly, and it's been really interesting.
So how has things changed? They've changed a lot in my discipline as well. When I was a graduate student, if you wanted to do a dissertation or a degree, you picked one pair of interactions and studied that really intensely. And the way we think about these interactions now is much more holistically and is depicted in this little picture behind me. And you can see it's pretty complicated, right? So when species interact, they don't do so in a vacuum. And the joint impacts of all the interactants, especially from a plant's perspective, shapes all of the traits and the responses of the plants to these interactions. So studying them in isolation was misleading because we were missing really what those joint impacts are likely to be. The other thing that's really, so it just makes the experiments that much more complicated. You have to layer on this many factors in order to tease apart all these effects. So they keep upping the ante with regard to experimental biology, especially field biology, which has always been challenging and is increasingly so. On top of it, though, our analytical tools for understanding these interactions have changed dramatically. We as humans have really poor senses of smell. It turns out most of the other organisms on the planet do not. And chemistry drives everything. So that's really been a revelation in my discipline, is how important chemistry, especially volatile chemistry, is for mediating these interactions. Most organisms in the world are traveling through smelling things rather than seeing things, and are using some of the senses we do. So our ability to trap minute amounts of chemicals in the air, analyze them with high-tech equipment, and understand what these chemicals are, how they're produced, has really changed pretty dramatically. The other thing is that my motivation for studying these systems was to go to the most pristine places and understand them in their natural habitats. But as we have worked for the last decades, it's been impossible to ignore the fact that climates are changing everywhere, right? And so understanding how all these interactions are simultaneously being shaped by human-caused environmental changes is really changed the discipline. And so my students, in particular here at GW, have really pulled me by my arm, into this field by coming to GW saying, I want to study climate change and how it affects these interactions, not just the interactions themselves. So they've really opened my eyes to that aspect of the research, which has really pushed my research in new directions. And that's part of, for me, that's part of the immense pleasure I get out of being a professor is learning from my students. So.
Yeah, definitely a lot of things have changed since I started my PhD. So, for example, this little device I'm holding in my hand, it's called a nanopore sequencing device. So this is roughly, just barely a little bit bigger than a USB drive, but this is a device that allows you to sequence massive volumes of sequencing data from DNA that you may be extracting from environments. So usually that's something you would do it back in the lab, but because of how portable this is, you can bring it to the field sites. You can basically in situ discover who's there, what kind of diversity you have. They have even used this, brought it up to International Space Station to do experiments to do sequencing in space. So this kind of thing I could have never imagined happening in my lifetime, but yet here we are. And also because of these advances in sequencing technology, we're generating massive volumes of sequencing data that's becoming computationally challenging to analyze. Back in the day, we would be just doing one organism or genome at a time, but now we are analyzing thousands of genomes of organisms from these environments. So we have to be a little bit more creative and learn a lot more new techniques like machine learning or big data type of analyses. And also we have to borrow other techniques from other fields like mass spectrometry or machine learning, like as you have said, to handle these massive volumes of data. Yeah.
I love hearing about the interaction with data science and machine learning, and I love hearing that the student-faculty interaction is pushing discovery. That's awesome. So here's another sort of fundamental question, and we'll come back down this way, starting with you, Jimmy, is how did you become interested in answering these questions?
Yes, definitely. So my interest in microbiology started with because I was really interested in how life originated in this universe. So, and then as you all know, Earth is the only example of life on this universe so far. So this is where we're gonna look at early life that may have originated here. So that's why I started microbiology as my major, because I started off with computer science major, and then I changed my major to microbiology. And then, yes, over the year, I became more and more interested in these extreme environments because they resemble conditions on Earth that are that you might expect to see in ancient times. So and if you go into those locations, you might expect to see some early or descendants of early ancient form that might be still persisting, and that's still true. So as I go more and more to these extreme environments, I'm starting to see more of these lineages with ancient features or gene signatures that I think are really interesting. So I keep digging for more and more of these lineages. So that's how I got started.
For me, I really got the bug, so to speak, as a first-year graduate student. I had studied biology and ecology as an undergraduate, not really knowing what I was going to do. I worked on plants as an undergraduate for my thesis, and I loved plants. But it wasn't until I was teaching a graduate course in ecology, and one of the lab exercises was I took the students out to an old field and we did sweep netting, which is this kind of crazy thing when we take nets and whack vegetation and collect all the little tiny bugs that are on them and kill them all, then bring them back to the lab and look at them under a microscope. And I knew almost nothing about insects at the time, but I had to help the students sort and ID them. And so I spent an entire weekend with the samples in the lab looking under the microscope. And I just couldn't get over how colorful and diverse and amazing these tiny little insects were under the microscope. They just sort of came alive once I got under the scope. And I was hooked. It was just, that was it. And I had to know more about them. And so that really is what led me to all of the rest of my work, is just sort of getting a visual hook of these crazy insects and how diverse and completely crazy they are. So.
Well, I got my start in marshes, tidal wetlands, and you don't really pick the marsh picks you, because most people find it smelly, buggy, hot, and muddy. But I seem to not have a reaction to poison ivy or mosquitoes, and I absolutely love these open landscapes. And I really like that they're this mix of sea and land. So I also think they're a really great system for experiments because you can move the plants around, you can manipulate a lot of the environment in this short stature plant community. So I got my start in marshes, and then I kind of moved upslope with sea level rise, I guess, because sea level rise is making forests convert to marsh. So forests, these coastal forests are now the marshes of the future. And honestly, I was very concerned and still am about the survival of tidal wetlands in this era of climate change because sea level rise poses such a threat to that ecosystem. So that motivates my work because one way that tidal marshes can actually survive is this migration pattern and the forest retreat that we see. So behind me is a new marsh where a forest used to stand. And we think that these are probably going to make up like the majority of marsh ecosystems by 2100 or so if we don't cross a tipping point where marshes can't actually survive.
It's always fun to hear why people take up the studies in the areas they do. So thank you for sharing that. So the next question is a bridge to the public. So we now know why you became interested in your questions. Why should the general public be interested in the questions that you study? And we'll take it back down the other way.
Thanks. Well, I guess my main reason that I think the public should be interested is because we all depend on the same ecosystems, right? There is no planet B. We're all living on the same Earth. And the ecosystems in the coastal areas, well, the 40% of the human population lives in the coastal zone. And so these are particularly important ecosystems to people. We depend on the services that ecosystems provide. And for example, tidal marshes are really important for carbon sequestration. And so a meter squared of tidal marsh stores 10 times the carbon that's stored in a temperate forest. So people think, about forests as carbon sinks, but these tidal marshes are really powerful carbon sinks. But they're also important for nutrient regulation, wildlife habitat, and people like them for recreation too, for kayaking, bird watching, and things like that. So I think that's a really important reason to care about these places, and that's what I tell the public.
Thanks. So insects that eat plants are the major pests in all of agriculture and forestry. So pretty much everything we study about their basic science has applied angles. In other words, direct applications to managing what are considered economically important pests in human systems. So those are really important. And we spent 50, 100 years using chemicals to just kill everything, basically, and we're now in an era where we're realizing that using natural enemies and other more sustainable ways of controlling pests are feasible if done correctly. And so coming up with utilizing nature's own control and regulation mechanisms is a much more efficient and much more environmentally sound mechanism. So we have to understand these interactions if we're going to harness their power as what we call biological control or pest management in some interesting way. The other thing is that plant-animal interactions are the basis for all of the amazing diversity of plant chemistry that has evolved in all the plants of the world, which basically form the basis for all of modern medicine, right? So the pharmacopoeia of chemicals that plants have evolved to defend themselves primarily against insects, we have used that, right, for our own health and benefit, and now are finding interesting and creative ways to study that chemistry and manipulate it for our own purposes. So medicine, agriculture, forestry, those are all impacted by plant-animal interactions.
Great. I think there's a next slide probably. So I know the extreme environments may seem like pretty far-fetched for us to be studying, and why should you care? But the reality is non-extreme habitats are also becoming subjected to extreme conditions, and we really don't know how microbes may respond to these extreme weather patterns or changes in the environment. So, for example, I can show you this. I wonder if you remember this incident a few years ago on the island of Maui. So there's apparently a pink phytoplankton bloom. It became like bright pink coloration, and then everybody was wondering what is happening there. And then so what happened that year was that there was extreme drought conditions that year, and so there was not a lot of fresh water input from the surrounding into this marsh area, like a wildlife refuge there. And so basically, because of that, the salinity of the pond increased rapidly, like 2-3 times the normal limits. And then that favors the growth of the certain bacteria that likes to thrive in high saline environments. So these are sort of extremophilic bacteria, but they are not really toxic to humans. They don't really produce toxicity. So, but general public was wondering, like, is it safe to go there? Can you actually touch the water? Yeah. So, but then, so that kind of questions I think that this kind of events are going to pop up more and more. And so I think that's why by understanding how microbes respond to these extreme conditions, I think we're better educated and learn how to respond appropriately. Yeah.
Well, you've sold me. The public needs to know, and that's why we're doing this. So another question is, so we know that you have made efforts to communicate your research to a broader community and to broader audiences, sometimes through publications in science and journals like that, but also through outreach to school children and through a variety of other media outlets. So can you talk with us about how and why you engage in those efforts and how you have overcome any barriers to communication? So I think the order this time is going to be John, then Karen, and then Jimmy.
Great. Thank you for that question. In my lab, science outreach is a critical component to training. So I've been engaged for the entire time I've been at GW doing outreach mostly with kids. I really like interacting with kids, and my wife is an environmental educator, so she drags me everywhere and sets everything up for me. And so in collaboration with my main colleague, who's a professor at Georgetown, we have developed a 20-year program, which we call the Caterpillar Roadshow, in which we collect and bring an entire zoo of locally collected caterpillars out to school children to teach them in small groups about the adaptations and biology of these insects that align with a lot of their science standards. So we basically pick a school every fall, usually in September, and I bring all of my students, and they don't really have a choice. They come and they really enjoy it. Some of them are here in the audience, I think, right now that are on this slide. And we do a really high energy, hands-on kind of activity to teach these kids about insect life cycles and some of the cool biology. And what we found is that having this up-close personal experience with these living organisms for kids that's on their level, these are organisms that they encounter in their daily life anyway. They see these things. And it's really just a sort of meaningful experience for us to learn how to talk about our science. So I view it as critically important for my graduate students in particular to learn how to talk to the public, and especially kids, about science. And it's really fun, too. It's really, really, really enjoyable. The other thing that we do is we do teacher training. So sometimes it's more effective. We can only reach so many kids. We usually do like the entire third grade of an elementary school or something like that when we do at one school. But what we found through the years is that if we can empower the teachers, then each one of them can reach a hell of a lot more kids, right? So we've done a series of teacher trainings, especially teaching DC public school teachers how to use things like iNaturalist and some of these apps so that they can then take those skills to teach their own kids. So I still like the kids, so I'm going to do the kids every year. But the teachers, I think, is a whole new angle that I think is really valuable. So, yeah.
Well, in my lab, we realized pretty early on that one of the most incredible things about ghost forests is that they allow you to visualize where sea level is rising. And that's a really powerful communication tool. So we thought, okay, let's see how we can kind of like use that to talk to people about climate change. And the way that we came up with, well, we were also facing a problem that we're studying the mid-Atlantic region, and we didn't know a whole lot about what's happening on the coasts of the Southeast and Florida, even though we can read the literature, but it's very, spotty geographically. And so we said, let's just like ask people if they know where the ghost forests are. So we developed this project called Ghosts of the Coast. That's a community science project. And luckily I had a great GW undergrad who was a double major in art and biology who could do a lot of design work and actually designed the website and designed some graphics and got really excited by this project, who took it to conferences but also to different universities and would kind of advertise the project in different university classes in the Southeast and the Northeast. And so we were able to collect a lot of observations. And the QR code on the slide here actually goes to our story map webpage, where if you ever spot a ghost forest, anyone in the audience, you can also contribute your observations. And I've used this even when I'm on trips in the South and I spot somewhere and I want to remember, oh, there's a ghost forest here, then I just add it to the database. So that's been a really nice way to engage and communicate with people, and it's really simple and something people can do like on a walk or a hike and kind of engage with nature that way. And then we have one other project that in the lab that's engaging the public or people in a different way around the science, and this is more at targeted stakeholders. So we have sea level rise is affecting these forests, it's affecting farmland too, and these are, lands that are really important to the property owners and to the industries that cut timber or grow crops. And so we're bringing all of the stakeholders involved in those sectors into meetings to talk about some of the solutions that we have, and we've developed like a whole menu of solutions, from using alternative crops that are more salt tolerant to planting perennial grasses that can tolerate the salt and provide other benefits, to pollinator wildflower mixes, to using the forest that's dying back as hunting land instead of timberland. So we're talking about these solutions with these stakeholder groups to find out more about what they prefer, and they are knowledge sharing about what's happening on their lands. And we're going to try to implement some of these solutions at scale. So just give you an example. One of these is coastal easements. So setting aside some of the property to not develop it while it's undergoing this kind of transition. And landowners can get a incentive payment for that. But we can also advise management practices like planting specific species, helping the marsh migrate onto those lands. So it's kind of a win-win solution for coastal property owners. And we're actually going to, we've had property owners approach our lab and my collaborators and say, we do not know what to do with our land that is changing so rapidly. Can you help us? Can you try some of these on our land? So we just have a new grant that this summer will implement a bunch of different solutions on a property in Crisfield, Maryland, to try to figure out what works and what doesn't.
Right. Yeah, I'm passionate about educating our next generation of scientists who are going to be next pioneers and leaders. So uh, I start with the immediate pool of students we have at GW, undergraduate students, and then I expand to the high school students around the DMV area, of course. I've been taking in a lot of students to work in my lab, not only for summer, but sometimes a little bit longer than that. But there's of course always barriers to communications, and as an introvert, I struggle with that a little bit to try to give out public speeches or to reach out to a larger pool of audience. But I try my best. But at this point, I would probably have to ramp up a little bit more of outreach activities to reach better, a wider range of audience around here, for sure, yeah.
All right, and our last question tonight is, so when you think about that broader communication, how do you decide who to target with your communication efforts, and what are you hoping to achieve? Do you think that policy formation and change can be influenced through science communication, the discussions about the work that you're doing? So why don't we start with you, Jimmy, and we'll work our way down.
Okay. Yeah, so I think there are many platforms that we can use to engage in a wider audience, of course. So what we've been doing recent years is like if we publish something, then we try to see if we can have some op-ed or opinion pieces of these articles that might reach wider audience. So for example, we published a paper in 2022 about these Hawaiian steam vent samples, and then even though it was a high-impact journal, because of these efforts, it kind of reached a wider audience, and then there were about 70 different news outlets that picked up on the stories, and then they reposted it to different types of languages and websites, so that was pretty interesting exercise to see, and I think that can continue to take place in our lab. And also, I've been experimenting with these social media platforms like Blue Sky or YouTube, for example, where I just started a lab channel where I'm going to be posting these field trips videos, as well as other things that might be of general interest to everyone. And also Wikipedia is also a pretty good resource, of course. And then that's where students in high school and undergraduate levels, they will probably use as a reference for their classroom. So these are all good sources to tap into. But
So one of the ways I think that's really effective to do science communication for my own work is to engage. I'm a DC native, so this is my home. I grew up here in Silver Spring, actually. And there are an amazing array of nature enthusiasts in the area. And most cities have them, and you have to find them, and they're not that hard to find, actually. And so giving public talks for non-specialists, I feel, is a really effective science communication. So natural history groups, birding groups, nature study groups are always eager to have speakers that are interested. So I try to take advantage of those opportunities. Another way is some really popular programs around the area are for retirees, so really educated people that retire from their jobs and are interested in nature study and things like that. There are opportunities for them to get certified themselves called master naturalist programs or master gardeners and all these kinds of programs that exist around urban areas. And so I've participated in those, giving lectures and field trips and things. Field trips is one of the best ways to do it, because I feel like when I'm in the field and can talk about these organisms and see them with people, that's the way to get them really excited. So any opportunity to go on a field trip, I'm in. So, and then in terms of policy and stakeholders, that as my colleague Dr. Gadan mentioned, in our work, some of the work we do, and because it has applied angles, especially with pest management, we have the opportunity to contribute sort of these not traditional science publications, but what we call extension publications. So, and I know Karen also has those kinds of opportunities, that for users, for people that are actually doing organic farming and things like that, they need fact sheets, they need guidance on how to better manage their organic farms to foster natural enemy control. And so those kinds of extension sheets, I try to get my students have that opportunity to write for this different kind of audience. And I think it's a really different way of thinking, and I'm still figuring it out myself, but I think it's an effective way to reach a different set of stakeholders that still could appreciate the science behind it.
Well, this question of like finding your audience for public communication, I guess maybe I didn't approach the question that way when I got into public communication, but rather I was looking for field sites and I needed to access a lot of private landowners' land to be able to understand like how are things changing here? Can I measure the salinity of your soil? Can I document the plants there? And they were very gracious about allowing me to work on their lands, and I've always been very respectful of that. And I just kind of approached it. I need to listen as much as I speak, because in rural areas, people who are working the land, they know a lot. And what I learned was like they're not like monolithic. There's such a diversity of opinions and knowledge, but you can really learn a lot in having open ears. And I think that has really served me well in terms of public communication, because you sort of build rapport, build relationships. It takes a long time, but you kind of start to engage some of the community organizations. There's land trusts and local NGOs and local chapters of national NGOs, and it sort of has ended up like building a really interesting network of stakeholders that has really given my research a new reach in the state and the region. So I think it's been very gratifying to me also, but just listening.
Well, this has been a fun conversation to listen to, so thank you. And now it's your chance to stop listening and to ask some questions. So I will be happy to take your questions. Yes?
If you could time travel to a certain period and do a field study then, what time period would you go to? I assume yours might be the very beginning of it all.
Yeah, roughly about 4 billion years old. Yes.
Jack,
yes,
do we
that's-
need to
that's-
repeat the questions? Okay, so if you can go back in time to a different time period, what time period would that be? To do a field study.
Yes, I would probably go back to roughly about 4 billion years ago when life originated, and I want to see where it started. But then back in those times, we won't have those technological advances to see what's there. Yeah, but that would be my choice. Yeah.
I love that question. I would definitely go to just after the last glacial maximum and see the salt marshes when they're forming as the glaciers recede, and that would be really interesting.
I guess I'm going to be the most current. So I would want to see what my field sites looked like pre-colonial times. So I'd really like to see, I'd like to show up in August or something in the height of the season and see what the diversity of insects was like before humans really changed things. So I'd love to see that. So.
Fascinating. Yes.
I guess my question, maybe mostly for Crystal, but probably all of you have an interesting answer to this. I noticed that you mentioned how your work has migrated, so it's being reported on in these newspapers and non-scientific journal settings. But do you have any concern about how your work might be misconstrued or misinterpreted outside the scientific setting?
All right, so the question is, do you have any concerns about how your work may be misconstrued through the more popular media?
Yes, absolutely. these popular or op-ed news outlet tend to either exaggerate a lot of the claims that we made or like completely twist things around. So sometimes it is just totally wrong. So this happens quite a lot with these new outlets, and then sometimes I see those as well, but then there's not a lot of things I can do to correct their mistakes. So for example, it was not in my lab, but one of my colleagues' lab, where they were studying these environmental chlamydia. So these are free-living chlamydia that don't cause STDs, but they were discovered in the deep oceans, somewhere in the Arctics, and then the news outlet, media outlet, twisted it to say like, if you swim in the Arctic Ocean, you will get chlamydia. It's that kind of thing. So it's really annoying, but it happens a lot. Yeah. So yes, I'm really concerned about how they really simplify things and then sometimes got the message all wrong. Yeah.
Well, my husband was a journalist for 10 years, so I have a lot of respect for the profession, I think, and I've worked with a lot of really excellent science journalists. So there's like, different flavors of journalists, and depending what kind of story, I think you need to be more careful or less careful. But I've found there's also like a professional science journalist who tends to work with me for like, it's not just a little interview. It's like, over the course of a week or 2 weeks or a month, they might do several interviews and exchanges over email. And can you fact check this, and do a really careful story. So I've also seen like kind of the opposite, where they do a really good job. And I just remember one of these, I'm blanking on her name. I wish I could remember right now, but she described our work. And then the intro paragraph described on America's first farmland. And like the Chesapeake Bay is this really important national estuary and historic estuary, but I had never thought about my work happening on like America's first farmland. I was like, wow, that's such a cool lens to put on the science, that these are like lands that have been worked for centuries. And so I really appreciate sometimes like that more general view.
Thank you. Yes, in the back.
Explain why people shouldn't care about global and how you will have to achieve it. My question is, how was that successful? So how much people are really care about global, and how people who are deciding environmental policies now they'll care about what they find.
All right, to summarize the question, so you've been engaged in outreach. How would you characterize the impact of that on people's interest, or whether they care about these issues, and the policymakers as well?
Yeah, that's a really good question. You can't win them all, but for me, I think reaching kids is to spark something, right? So it doesn't take much for a kid to just get excited. So I've watched it happen I've talked to teachers, and it's like sometimes just one encounter, where their imagination is sparked in such a way means that they're going to do something like that. And I had that same experience as a young child. So I had a great science teacher who, once I was I was in 7th grade. I knew what I was going to study in 7th grade, So like those things can happen. They don't always happen, but even really beyond that, I think just giving people, kids or otherwise, or adults, and a greater appreciation for nature might make them think a little bit more. And I tell my students that I really care more about how you vote, right, than understanding what the details of the Krebs cycle or something like that, right? We want to make good citizens out of people, and science education is part of being a good citizen. So that's kind of the way I approach it.
Yeah, so a lot of our research may seem a little bit esoteric and like, okay, we are really interested in these topics, but then The general public don't see it that way, not necessarily, but if we can make a connection to how their day-to-day life works and how is it really relevant to their way of life, I think that can actually persuade the general public to be a little bit more interested in our research. I try to make that connection in my classroom at least, and I try to teach students like, okay, why should you care about diversity, microbial diversity, and then why it wouldn't make sense if you become a physician and by not understanding the diverse microbial life you have in this planet, you cannot really be a good physician. So that's the kind of message I try to drive home in my student at least. Yeah.
Right. Any other questions? Yes.
I really enjoyed the discussion hearing you speak about your work. I was wondering what you're looking forward to in 2026.
All right. So what are you looking forward to in 2026?
I'll start. I cannot wait for the spring flowers to start blooming. So spring ephemerals are going to be popping in late March, probably. So for me, part of what's so great about being a field biologist in the temperate zone is by the time you're done and sick of field work and all your bug bites are, bothering you, I do get bug bites. It ends, and then you have this long, dark period here in the middle, and then I just cannot wait for the new field season to start every single year, and the whole cycle starts again, and it's like a kid just getting to observe that things coming to life early on. So I teach a class on plant-animal interactions, and day one, the first plant, we did plant of the day. So our plant of the day on day one of the first lecture was the eastern skunk cabbage, which is this amazing thermogenic plant that generates its own heat and flowers in January. So it's flowering right now. So I was like, we don't need to wait. We can go out right now, to the field. Let's go look at skunk cabbages. So I think that what I look forward to every year is a new field season, new discoveries that my students are going to make because they do every single year. Something just gobsmacks me every single summer that my own students discover, and both my undergraduates and my grad students. So I get to witness that. So that's the best part of my job.
So a lot of the science that I do is long-term science, like sea level rise, very incremental, you got to watch a long time. And I'm involved in a National Science Foundation project that's called Long-Term Ecological Research. So this is a place that has had continuous funding since 1987 and has been able to take the same measurements year after year after year. And we've been studying this forest now for 7 years. That's part of this field site. And we started a long-term experiment in November of 2024, which maybe now sounds like a long time ago, but because these are trees, they respond very slowly. And this year is the year where we're really going to see change happening in that experiment that I've been working so long to plan and lead up to. So I'm really excited to see what happens because finally it's a disturbance experiment. So we killed trees, places to try to understand this transition and how it happens across an environmental gradient. And we're finally going to see that transition occur in real time. Plus, I think my lab right now has kind of like this special sauce. Like I have students with really amazing computational abilities, with really amazing statistical abilities, and we've been collecting all these data sets, and each one of us keeps saying, I think we're on the verge of a breakthrough. We have all the pieces of the puzzle, and I think 2026 might be the year where we put the last piece in. And plus, I just hired this really great research technician. So it's going to be a great year.
Yes, I definitely second John and Karen's sentiments. And I look forward to field season as well. So especially around summertime when we're not teaching, then I get to go to these interesting field sites. And I'm currently involved in a project to investigate these hot springs from very like unknown locations, so outside of Yellowstone or any national park system that are poorly preserved and protected. So I'm hoping to explore these never before seen hot springs that are only in databases so far, but then I'm gonna go and see if they are there and then actually collect samples from those locations. So I look forward to the field season in the summer and then to bring my students and then show them how things are like, yeah.
Well, I think that's a great way to end the evening. Thank you for your questions. Thank you to the panel for sharing your expertise with us. It's been a really wonderful conversation, so thank you. I also want to thank some people who helped make this possible. So here in the room filming us from SMPA, we have Chris and Jack, so thank you for your work on that. And I want to thank Beth and Max from the Columbian College office for helping put this together, as well as Associate Dean, easy for me to say, Associate Dean Evie Downey, who helped organize this as well. So please join me in thanking the panel for your wonderful conversation. But the conversation doesn't have to end. We have a reception waiting outside those doors. So come and join us and continue the conversation. Thank you all. Thank you. That was great. I really enjoyed that. And where do I find a skunk?
Any low-lying area. Those aren't, these don't look like